Welcome everyone to the lab house. Today we will discuss about the DNA denaturation and renaturation. In the previous video we discussed the molecular structure of DNA. We see that the DNA contains major groove and minor groove, the rise of 0.34 nanometer and the pitch, which is also called helical periodicity, of about 3.4 nanometer. The diameter is about 2 nanometer. Then we saw the components of DNA: deoxyribose sugar, phosphate group, and nitrogenous bases. We see the difference between nucleoside and nucleotide. Nucleoside contains sugar and base only, while nucleotide contains the phosphate group too. Then we see the nucleotide formation. There are two covalent bonds that are formed between sugar and base called glycosidic bond, and between sugar and phosphoric acid called the phosphodiester bond. The phosphodiester bond or the phosphodiester linkage is formed between the 3-hydroxyl group of one sugar and the 5-hydroxyl group of the next sugar, and this process is also known as concatenation. Then we discuss the base pairing rule, the hydrogen bonds. There are two hydrogen bonds between adenine and thymine and three hydrogen bonds between guanine and cytosine, making the guanine and cytosine more stable. And the melting temperature depends upon the guanine cytosine stability. Higher the GC content, higher will be the melting temperature. Then we saw the tautomeric forms of the bases. A and C exist in the minor forms mainly, while G and T exist in the keto form mainly. We discussed the mnemonic for the amino form AC and AMI. Adenine and cytosine exist in a minor form predominantly. Now let's discuss the DNA denaturation first. As you can see, on heating, the double-stranded DNA are converted into two single strands. This is known as denaturation, breaking the hydrogen bonds of DNA. And on cooling, the single-stranded will rejoin again to form the double strand. This is called renaturation or hybridization. So DNA denaturation is basically breaking of hydrogen bonds. It is done by three chemical methods: heating, as you have seen in the previous picture, sodium hydroxide treatment, and salt induction method. Heat denaturation. Heating of DNA above 95 degrees Celsius will causes DNA strands to unwind and separate, known as melting, and on cooling leads to possible renaturation or reannealing or hybridization. It is commonly used in PCR. It is a simple method but less accurate and less precise. The more accurate method is the sodium hydroxide method. Sodium hydroxide will break the hydrogen bonds. It is a rapid denaturation method, less than 1 minute. It is controlled by sodium hydroxide concentration. Less sodium hydroxide, more time it will take. And renaturation occurs by phosphate buffer. In the previous method, renaturation occurs by cooling. It is an accurate and precise method, but it requires careful chemical handling. The salt induction method. Salt induction method uses high salt concentration combined with acids, heat, or alcohol. Remember that only high salt concentration causes renaturation, while high salt concentration combined with acid, heat, or alcohol will cause denaturation. And this kind of denaturation is irreversible. It will permanently disrupt the DNA structure. In case of high salt concentration alone, renaturation occurs because cations in the salt will neutralize the negative charge in DNA. As we know that the single strand DNA are negatively charged because of the phosphate backbone. So the two single strands will repel each other, so no rejoining can take place. But in case of high salt concentration, the cations, positive ions present in salt, will neutralize the negative charge. So so there will be no repulsion and the DNA strands can rejoin again. The salt induction denaturation method is used for large scale DNA processing when large amount of DNA is required, for example in DNA extraction or DNA purification techniques. Applications of denaturation. Denaturation occurs in PCR to separate the DNA strands for amplification purposes. In DNA sequencing to read the nucleotide sequences in a single DNA strand. Molecular cloning to insert the DNA from a single strand into vectors plasmid for cloning purposes. And Southern blotting. Remember that Southern blotting involves both denaturation and renaturation. It is similar to Western blotting. In denaturation, the strands will get separated, and due to electrophoresis, we will get fragments of DNA, and the fragments will get transferred to a membrane, and fluorescent or radioactively labeled DNA probe is added to the fragments, and both the fragment and the DNA probe will rejoin, known as hybridization or renaturation, and the probe will detect the target sequence on the DNA sample. Because probe is a single stranded sequence of DNA that is used to identify specific target sequence on the DNA or RNA present in the sample. So in the DNA fragments, the specific target is detected by the DNA probe by rejoining them. So denaturation occurs at last and denaturation occurs first. That's all about denaturation. We will discuss renaturation in the next video.